In this video, I will talk about RT-PCR, starting with the process of reverse transcription in retroviruses. So, during reverse transcription, viral RNA is converted into DNA, which is then integrated into the host genome. The enzyme responsible for conversion of viral RNA into the DNA is called reverse transcriptase. And in fact, it is RNA-dependent DNA polymerase, because based on the viral RNA, the DNA molecule is synthesized. Once we have the integration of the newly synthesized DNA into the host genome, it allows the virus to replicate, and new virions with RNA genome are produced. So here we have the production of RNA viral genome. And additionally, the viral RNA serves as a template for the synthesis of viral proteins. And this reverse transcription is widely applied in molecular biology for RT-PCR, so reverse transcription PCR, to synthesize complementary cDNA from RNA template. And in a moment I will explain how it works and what's the purpose of this RT-PCR reaction. In the laboratory, RNA cannot be directly amplified using PCR reaction. Therefore, to detect the presence of RNA, most commonly we are interested in mRNA, this molecule must be first converted into the DNA through the process of reverse transcription. In fact, it is converted to so-called copy DNA, cDNA. And conversion and obtaining cDNA allows us to amplify and analyze the copy DNA using PCR reaction. And on the slide you can see how exactly the reverse transcription procedure looks like in the laboratory. So here we have the mRNA molecule, and to reverse transcribe this mRNA molecule into the copy DNA, we need the enzyme reverse transcriptase, originally isolated from retroviruses, and now it is used as recombinant protein. But apart from the enzyme responsible for this reverse transcription, we need the starter. And the starter, it is a universal one in the process of reverse transcription. It is composed of the thymine deoxyribonucleotides. So it's in fact the short fragment of DNA composed of thymine deoxyribonucleotides, and it will bind to poly-A tail, which is present at 3' end of mRNA molecules. And once the primer oligo-DT is attached, the reverse transcriptase, being RNA-dependent DNA polymerase, can start to extend this starter and, in fact, reverse transcribe RNA into the DNA. It's important to keep in mind that every DNA polymerase, and reverse transcriptase is definitely DNA polymerase as it synthesizes DNA, requires starter to initiate the reaction. And once the reaction is completed and RNA is reverse transcribed into the copy DNA, RNA molecule is degraded, and now we have the copy DNA being like the mirror of mRNA, and this copy DNA can be used as a template in PCR reaction. So now we can explain what RT-PCR means. RT-PCR stands for reverse transcription polymerase chain reaction, and it combines reverse transcription, where RNA is converted into the complementary DNA, copy DNA, so cDNA, and subsequently the PCR reaction is performed using cDNA as a template, which is amplified in PCR reaction. And very important issue, RT-PCR is a method for studying gene expression because when a gene is expressed, based on the gene sequence, RNA molecule is produced. Later on, this RNA, if it is mRNA, can serve as a template for protein synthesis. And if the RNA is produced in RT-PCR, it can be easily converted into the complementary copy DNA and detected in PCR reaction. So definitely RT-PCR is a method to analyze gene expression. If we ask the question, is the gene expressed? So is the RNA molecule produced based on the gene sequence? We will apply the RT-PCR method. And once cDNA is synthesized, it is single-stranded DNA molecule. So in the first cycle of PCR reaction, only forward primer can find the complementary sequence. Of course, primer will be specific for particular cDNA we are interested in. And after the first cycle, new strand based on the forward primer is synthesized. And in the second cycle, we have already the template for the reverse primer, and then amplification looks like exactly the same as we discussed during PCR procedure. And after PCR reaction, when approximately 30 cycles are completed, we may perform agarose gel electrophoresis in the presence of, for example, ethidium bromide to visualize the presence of the RT-PCR product. And in this situation, the presence of the product means that the particular gene was expressed because we detected amplified cDNA, which was formed based on the RNA formed based on the sequence of particular gene. And now let's apply our knowledge into the practice. We have the question, does the amount of the mRNA for the gene encoding carbonic anhydrase 9 change in hypoxic condition? When the cells are cultured in hypoxic condition, of course they produce lactate and the pH drops, and as a consequence they need to cope with this dropping pH, and the enzyme carbonic anhydrase 9 helps them to manage with this low pH, being the consequence of deprived oxygen environment. So to answer this question, of course, the RT-PCR procedure is performed. Initially, the RNA is isolated from control cells and cells cultured in the oxygen-deprived environment. RNA is converted into the cDNA, total mRNA is converted into the cDNA, and later on the cDNA is amplified with the primers specific for carbonic anhydrase 9, so specific for cDNA of carbonic anhydrase 9. And once the PCR is completed, we have gel electrophoresis and we can observe the product, control cells, here we can see the product, and cells cultured in oxygen-deprived environment, and we can see much thicker band on the gel. So it means that in the cells cultured under hypoxia, oxygen-deprived environment, higher levels of mRNA for carbonic anhydrase 9 were produced, and it resulted in the production of more cDNA during reverse transcription, more cDNA particularly for carbonic anhydrase 9, and it led to the formation of more PCR product once the primers were dedicated for cDNA of carbonic anhydrase 9. If we have more PCR product, obtained in PCR reaction, we can see thicker band on the gel as presented in our example. So thank you very much for your attention, and it was the short story about RT-PCR reaction and its application in molecular biology.